Chapter 6, Lipids Screencast 4. Now, this particular screencast is going to be focused on the transportation of lipids in the blood. And this is a relatively complex topic. So this might be something that you need to look at again. There's going to be a lot of terminology that's going to be introduced here, especially a lot that will be looked at later with regards to health implications of lipid, heart disease, and so on. So some of this we will actually be revisiting. Now, when we look at how lipid is going to be transported in blood, we need to remember that we have a pretty significant challenge, as water and oil and anything that's lipid in nature do not mix. So that creates a pretty significant problem. Now, lipids will be transported in the blood as lipoproteins, and there are a number of these lipoproteins that function in this particular manner. So the ones that you need to be comfortable with are going to be chylomicrons, very low density lipoproteins, intermediate density lipoproteins, low density lipoproteins, and high density lipoproteins. Now, quite often you may hear of low density lipoprotein referred to as bad cholesterol and high density lipoprotein referred to as good cholesterol. It's not. We're talking about the carrier of the lipid. So that's something that I want us to really be comfortable with. Now, lipoproteins are actually composed of a core of lipids, and they are covered with a shell of proteins, phospholipids, and cholesterol, and this is how they manage to move in an aqueous environment. Now, when we look at the following, so you should be able to identify the type of lipoprotein, its primary component, and its key role. So when we talk about the first, or chylomicron, the primary component here is triglyceride, and it is carrying dietary fat from the small intestine to cells. VLDL is primarily made up of triglyceride, and this carries lipids both taken up and made by the liver to cells. LDL, this is primarily made up of cholesterol, and this is carrying cholesterol made by the liver and from other sources to cells. And then we have HDL, the primary component is protein, and that actually helps remove cholesterol from cells and in turn excretes cholesterol from the body. So we can see the positive function associated with HDL. Now here we're looking at an overall diagram that's focusing on the structure and composition of the various types of lipoproteins. So first, if we look at the chylomicron, and you can see it's 80 to 90% triglyceride, very, very low density, not a lot of cholesterol or phospholipid, and hardly any protein at all. Then we move on to the VLDL, and once again we see something that's primarily made up of triglyceride, a little bit more cholesterol than what we saw in the chylomicron, a little bit more phospholipid, and a little bit more protein. Now, when we look at LDL, notice here this is primarily made up of cholesterol and phospholipid and a little bit of protein. And then we have HDL, and notice that's primarily made up of protein. So we can see, so you should be comfortable, not with the exact percentages, but with the primary makeup of each of these lipoproteins. Now, when we look at the interactions of the lipoproteins, there's a couple of things that I want to keep in mind. So the first thing that I'm going to do is kind of give us a run through of the actual interaction. So the first interaction is chylomicrons carry absorbed fat to body cells. Then, if we look at the green or 2, VLDL carries fat taken up from the bloodstream by the liver, as well as any fat made by the liver to body cells. 3, we look at the LDL, arises from VLDL and carries mostly cholesterol to cells. And then 4, the HDL arises from body cells, mostly in the liver and intestine as well. So let's kind of look at the actual pathways and interactions here. So here we see the chylomicron, and there's basically 3 things that are happening here. So absorbed fat from the intestine is actually traveling as chylomicron, and the fat is making its way to body cells after breakdown by lipoprotein lipase. So we can see that here. And then what's left over the chylomicron remnant moves on to the liver. So we can actually see that interaction. So 2, we're actually looking fat that's leaving the liver, and that travels as VLDL. Now, the fat moves on to the body cells, and it's going to be moving there through lipoprotein lipase. And what we have now is another remnant. That remnant may also called IDL or intermediate lipoprotein, and that is going to move on to the liver. Now, as cells remove fat from VLDL, this is how we actually form LDL. So that's that last phase here. LDL is going to be taken up by receptor pathways in the body cell. And then what we're going to look at, we'll talk about this a little bit later, as far as the scavenger pathway in blood vessels. Here we see oxidized LDL, and here's where we start to see the risk factor of the cholesterol that will potentially form plaque. Now, if we look at the HDL, that comes from the liver, and what it does, it's going to bud off of other lipoproteins. And what happens here, notice what it does. It basically picks up cholesterol from the body, and then it's going to take it to the liver for disposal. So very complicated. And notice we just don't talk about one. We're looking at chylomicron, we're looking at VLDL, LDL, HDL, and so on. So this is a relatively complex pathway and something that you should be comfortable with. Now, when we look at chylomicrons, and as I indicated before, dietary triglycerides that's reformed in the absorptive cells of the small intestine. So here we're going to see cholesterol and phospholipids, but mostly triglycerides, and the shell actually consists of phospholipid, cholesterol, and protein. Now, apolipoproteins, these are found in the protein portion of the shell, and what they do is they're responsible for turning on lipid transfer enzymes. Now, they're also going to assist in binding a lipoprotein to a particular receptor, also assist enzymes. Now, chylomicrons are going to enter the lymphatic system via lacteals, and they travel through the lymphatic system to the bloodstream and enter at the left subclavian vein, and nutrients are then transported to body tissues via that pathway. Now, when we look at lipoprotein lipase, this is an enzyme attached to inside of cell membranes, and it's activated by one of the apolipoproteins. What it's going to do, it's going to hydrolyze the triglycerides from chylomicrons, and then free fatty acids are actually taken up by the cells, repackaged into triglycerides, and used for energy or put into storage. What's left after this is the chylomicron remnant. Now, the liver can actually make fat and cholesterol from carbohydrates, protein, and free fatty acids. So the liver will coat the cholesterol and triglycerides with the shell of protein and lipids, and this is how very low density lipoprotein is formed, and that will enter the bloodstream. Now, lipoprotein lipase will hydrolyze the triglyceride that's in the VLDL. We see the free fatty acids entering the cells, and what's created is intermediate density lipoprotein as the triglycerides leave. Now, the intermediate density lipoproteins lose more triglyceride by an enzyme called hepatic triglyceride lipase. So this is what now creates the low density lipoproteins, which are composed mostly of cholesterol. Now, one thing that I want us to keep in mind here is there will be questions about these receptors and so on. The receptor pathway for cholesterol uptake. And so what happens basically removes LDL from blood by cells with LDL receptors that are called B100. Now, the LDL is broken down to cholesterol and protein once in the cell. Now, if free cholesterol saturates the cell, then that B100 receptor stops taking LDL from blood. This is how we start to see the concentration of LDL actually increasing in the blood, And here's one of the issues that happens. This LDL can be actually oxidized by free radicals. Antioxidants can help in this process, so it's important that we are providing our body with adequate antioxidants through many of the vitamins. Now, the scavenger pathway for cholesterol, what this does, it uptake, collects, and degrades extra LDL in blood. So what happens here is these macrophages, they detect, engulf, and digest LDL. Now, the cholesterol builds up in these macrophages and it kills them. Now, these cholesterol-filled macrophages build up on the inner blood vessel, and that is the actual development of plaque. And there's a progression here. So we have the original progression of the plaque formation. This actually becomes atherosclerosis, and that eventually can potentially become an occlusion and create a blockage somewhere. If it's in the heart, then we're looking at a myocardial infarct or a heart attack. Now, diets rich in saturated fats, trans fats, and cholesterol actually encourage this process. So that's why there's such an emphasis on these particular factors. Now here, if we look at the actual receptor pathway, we can see a couple of things that are going on here. So we see that beta 100 receptor, and we can see the LDL. And notice there's the appropriate lipoprotein to bind to the receptor. So these cells have these pits on the surfaces where the receptors can actually interact with the LDL, and then that actually allows it to be carried into the cell. So the LDL binds to the LDL receptor in the pits. Now, here we can see the LDL bound to LDL receptors is taken into the cell. Now, as we noted before, once that cell becomes full of cholesterol, then we're going to see that route of plaque formation. Now, here we can actually see the progression of atherosclerosis, and we have a nice clean vessel. Now, one of the things that I want to point out here is it's not until a blockage is close to 90% or greater where you're actually starting to experience symptoms. So by the time someone experiences symptoms, the disease is very well progressed. So I want to make sure that is something that we kind of remember. There's actually been a number of studies. There was one done on soldiers that were killed in Vietnam. And these were relatively young men, because remember, there was still a draft then. They were on the front line, so we're talking about young, relatively healthy. And what they did was they looked at the coronary arteries of these soldiers that were killed, and already at a very early age, they were demonstrating blockages of 50% and greater. Okay, so 18 to 20-year-old men already walking around with blockages of 50% in their coronary arteries. So then here we can actually see early injury, and that's one of the things that actually promotes this process, and it's damage to the inner layer of the blood vessel. So we can start to see the progression and the beginning of that cholesterol plaque. Then we start to see the advancement of the plaque, and notice the impact it has on the vessel. So that vessel is now getting pretty occluded, and eventually we get to the point where we have a complete blockage and we have a clot that may actually break off and create an issue elsewhere. Now, HDL, this is actually high-density lipoprotein, and quite often when you go in and get a lipid profile, it is referred to as the good cholesterol. It has the most protein of any of the lipoproteins. It's produced by the liver and the intestine, and what it does, it actually roams the bloodstream, and it actually picks up the cholesterol from cells that are dying or other sources. Now, this cholesterol is going to be donated to other lipoproteins and will be transported back to the liver for excretion. Now, the other thing that HDL does, it helps to block the oxidation of LDL, so we know that it actually slows the development of cardiovascular disease.